Okay, so beginning our discussion, we're going to talk about post-transcriptional regulation of gene expression, and a lot of this has to do with differentiating that RNA processing. So you'll remember, of course, when we talked about gene expression, we talk about transcription and we talk about translation, and in between the two, we talk about what? What happens in between initiation, in between transcription and initiation of translation? Yeah, we have the spliceosome cuts out introns, we add that guanine cap, we add a poly A tail, we process that RNA. Because after transcription in eukaryotes, that RNA is not ready to be translated. It has to be processed. So the question is, well, what can a cell do during that process that is going to regulate the expression of whatever the protein is that would be translated by that mRNA? And so RNA polymerase II is our specific machine that transcribes mRNA. And here, the mRNA now is exported to the cytoplasm, and then here we've got some processes that we can actually modify this in different ways. Now, this right here, this RNA, the miRNA, this is one of those small interfering RNAs. One of those RNAs that can go in and interfere with our other mRNA before it gets translated. So if you go all the way down here, you can see that here is our pre-miRNA all the way until it's been fully processed. And do you see what it's doing here? Might be difficult to see, but do you see what it's doing here? When you see this, what process is happening here? This is translation. And so this miRNA is actually physically attached to the mRNA, and so it's preventing the translation of that miRNA. On top of that, we already know this. Some of these we've talked about before. We can splice out different sections of the pre-mRNA to produce different mRNAs for translation. And we know that the site of the cutting and the site of the splicing is determined by regulatory proteins. And so those regulatory proteins are cell specific. So the same gene will be cut differently in a liver cell than it would be in an epithelial cell. Okay, because those cells are transcribing different regulatory proteins. On top of that, we've got in the 5' untranslated region, which is before the part that actually gets translated, there are modifications that we can do there to shorten the half-life of that RNA. And the half-life is basically how much time passes for half of it to be degraded. Okay? And so most often you use half-life in a chemistry application, right? Which is why you're all like, oh, I love half-life, right? Because y'all have done like radiochemistry. Yeah, oh yeah. And so, but even these mRNAs, they have a half-life, and so we can modify these at that 5' untranslated region to make it to where they don't last as long. Okay? So if you don't want as much expression of an mRNA for some reason, you make it to where that mRNA is not going to stick around as long. So it's not going to be translated as many times. Not going to be translated as many times. Okay, the poly A tail basically does the same. The longer the tail, the longer this mRNA survives. So if you want, again, you want to transcribe this mRNA and you want to translate it, but you don't want to make a lot of that protein. So what do? You put a very short poly A tail on it, and you shorten that 5' untranslated region. And it makes it to where that mRNA gets degraded more quickly, so you won't make as much of that protein product. And then what we just saw in the previous diagram our cells can transcribe an miRNA that is complementary to your mRNA. We got some markers that work in here. I want to show you something. I think you're going to like this. Okay, let's say this is our DNA sequence that we are going to transcribe. Okay, so we are going to transcribe this DNA sequence. It is ATTGCA. Okay, and so if we transcribe this into RNA, let me write transcription above here. So we transcribe this, what does it become? What is adenine when it's in RNA? It's still adenine. What is thymine? U, it's uracil, G, C, A. Okay, so that's what it looks like when we've transcribed this sequence into RNA. Okay? And so what if we do this? We transcribe this, we process it, but then the cell receives a signal that basically says, don't do that, right? Don't actually follow through on that. What does the cell do? It's already transcribed it, it's already properly processed it, and maybe it put a long poly A tail on it. So it's going to survive for a long time. Yeah?
So in the transcription, does it transcribe the opposite half and then transcribe it? I'm confused about the order of transcription because I thought it was the opposite.
Well, it's got to read the opposite to transcribe this. But we're basically, okay. This is what we want to copy, but we want to copy it in RNA, in the RNA letters. But yeah, if you were actually going to get this, RNA polymerase will have to read the opposite strand to write this, but this is what it's copying. So, but you copy by reading the complement. Okay? What's the complementary strand to this? So DNA is double-stranded, right? So if we want to copy this, this is what it would look like as an RNA, right? And now this is our messenger RNA. One of these was working better this morning. That's okay. Okay, so here's our messenger RNA. We've already made it, but now we want to regulate this. So what is the complementary strand to this in the DNA? Because DNA is double-stranded, what is the other strand? If this is A, what's here? T. If this is T, what's here? Wow, that one's even worse. If this is A, this is T. If this is T, what is this? A, C, G, T. Okay? That make sense? And so what do we do if we've already transcribed this? We transcribe the complement. So if you get an RNA version of this, instead of a T, what would we have? We'd have a U right here. The A's would still be A's. The C would still be a C. The G would still be a G. And this last T would be a U. Okay? So we transcribed the complement. But now look at this. What is U complementary to? A. What's in this position? A. What is A complementary to? U. A is complementary to U. C is complementary to G. G is complementary to C. U is complementary to A. We just made another RNA. This is our miRNA. That is perfectly complementary to this. So we make this and we release it, and it's going to go out-compete ribosomes for this. And so translation will never happen.
What did you say the name of this process was?
It's just all post-transcriptional regulation. Yeah, post-transcriptional regulation. Yeah.
How does it outcompete like the mRNA?
Because it matches it at better. So you remember, during the process of translation, you're bringing in one anticodon at a time. Right? The tRNAs are coming in one at a time, matching 3 of the bases. But this perfectly complementary miRNA might match this at 100 bases. And so it gets to it before all of the tRNAs can get there. And it basically gets there, and like in the image, which we'll see again, the image, it gets there and it just sits there. It's bound to that mRNA that you made, and it just sits there. And so you can't read it. The tRNAs can't read it because the codons aren't accessible. They're covered by a perfectly complementary piece of miRNA. Does that make sense? And so this is kind of bridging that boundary between post-transcriptional regulation, which is all having to do with RNA modification, and the translational regulation, where now we're interfering, we're blocking that mRNA from being translated. Okay? And then that's if you were asked the question of what is going on during the regulation of gene expression at the level of translation, it's all about this. Having RNAs basically interfering with the translation by binding to a complementary mRNA. And so the rest of what we're going to discuss is all going to be, well, what happens if we've already made the protein, right? So we failed to regulate it at the level of transcription. We already transcribed it. We failed to regulate it at the level of post-transcriptional. We already modified it correctly and sent it out. We failed to regulate it at the point of translation. It's already been translated. The protein's already been made. What do we do now? What do we do now? Now, some of this you should be able to predict already from our transduction cascade. What do some proteins need in order to be activated? Well, kinase enzyme that will phosphorylate them, right? And so some proteins can be turned off by removing phosphate groups. Okay, so the last question we're going to deal with is this. Well, how are proteins regulated? What are some of our post-translational regulatory processes? Yeah.
So post-transcription, translation,
and then post-translation.
Are those all outside the gene?
Yes. Yep. Oh, absolutely. And these are all only found in eukaryotes. So again, prokaryotes can only regulate gene expression at the level of transcription. Okay? Eukaryotes can regulate at the level of transcription, but they can also do it at the level of transcription above the gene, which prokaryotes can't do because prokaryotes don't have the kind of chromosomes that eukaryotes have. And then prokaryotes can't do anything else. They can't do post-transcriptional, they can't do translational, they can't do post-translational. Okay? So here's probably the most permanent option. So you have a protein, you've already made it, but you don't want it anymore. So as a cell, the best thing you can do is to tag this for destruction. And so there's a small protein called ubiquitin that when you tag another protein with ubiquitin, it basically labels it as this needs to be destroyed. And then other proteins will see this tag, will grab this protein, and will take it to a proteasome, which is a organelle that unfolds and destroys proteins. And see this proteasome just chews it up into individual or into small complexes of amino acids that can be processed. And then these amino acids can be recycled. So this is the same thing you do with like protein in your diet. It's foreign, and so it has a label that it needs to be destroyed. It'll be taken to one of these, it'll get destroyed, and you'll use those amino acids. But this is also what your cells do when they've already made a protein that they don't need anymore and they don't want around. Because proteins are doing work, right? They're doing things. And so if that needs to stop as the cell responds to some signal, you need to get rid of that protein, okay? And so you mark it for destruction. So all of these happen after the process of translation. So these are ways in which we can modify proteins. We can chemically modify them. For example, our transduction cascade gives you a wonderful example of how you can chemically modify proteins. There, we are chemically modifying them by turning them on. We're activating proteins that are dormant, that were synthesized in an inactive state, okay, by phosphorylating them. But you can, if you have now an active form of that enzyme, how do you deactivate it? You remove the phosphate group, okay? Enzymes called phosphatases do that, which literally means to cut a phosphate, okay? the proteins can be processed. And so many of the proteins in our cells are translated and folded, but are still inactive until a piece is cut off of them. Until a piece is cut off of them. I was going to say it's like the lancets we used in lab yesterday, but not all of you were in lab yesterday. There's a little plastic cover over the point at the end. Okay, and the lancet is useless until you remove that little plastic cover. In the same way, some of these proteins are useless. They don't carry out their activity unless you remove a piece of them. Okay, and then what we just saw in the previous slide, the proteins can be permanently destroyed. Okay, they can be marked for destruction, taken to a proteasome, and chewed up, recycled. And so these are all post-translational, and these do not happen in prokaryotic cells. Okay? Any questions? Okay. The last thing I want to do is, as we work together for the very last time, is just to put this in the context of what happens through the process of development. Because it's one thing to say eukaryotes, we can have division of labor. Like we can have liver cells, and we can have epithelial cells, and we can have kidney cells, and neurons, and cardiac muscle cells. But we need to talk about, okay, well, what happens during development to basically control what the cell becomes? What happens during development to control what the cell becomes? No questions? Okay. I understand. It's just like, Dr. Engel, just finish the material. Let us go. Get it. So what are the processes of development? What are the processes of development? Well, for a multicellular organism, you begin life as a zygote, and your process of development will continue until you reach adulthood, which is defined as development is done. Okay? You have reached adulthood when development is done. Development has finished its process. So it requires several things. First, a lot of mitotic cell divisions. So we talked about mitosis for exam 2, but one of your study guide questions for our final exam asked you, how does meiosis differ from mitosis? Bringing back some of what's happening in mitosis. On top of that, we have to talk about it again. And so what is the product of mitosis? Or what are the products of mitosis, rather? Two diploid, genetically identical cells. And so why do we need a lot of that? Well, the zygote is a single cell. And if you are going to make a multicellular organism, you need more than a single cell, right? And if you're going to have a good multicellular organism, those cells should be pretty much genetically identical. And so that's what mitosis does for us. So as the zygote does this, there are materials that make sure every cell gets the right amount of cytoplasm. So some cells are going to always be big. Some cells are going to always be small. And so while mitosis, you have to remember, mitosis is the division of the nucleus. Cytokinesis is the division of the cell. And cytokinesis does not always separate the two genetically identical cells into the same size. Okay, sometimes that division is asymmetrical. We also need to move cells around. And so you see this when you get to certain points of development, they are basically noted by the shape of structures that cells form. Cells will start to move inward to form inner layers. Some cells will start to move outward to form the outer layers. You get a process of what's called induction. And so in here, you basically have a group of cells called the inducers that tell the surrounding cells what they need to become. Okay? What they need to become. And those cells are called the responders. And this seems to be the most important process in all of development to control what that cell becomes. Its external environment tells it what to become, which is really interesting for animals, at least, and some other eukaryotes, is you can remove a cell from that embryo, and now its environment is not telling it what to become, and it starts the process over. And so this is how you get identical twins in animals. That at an early stage in development, a cell is removed from the embryo, and its environment now is not telling that cell to become something, and so it starts the process over and forms a new individual. But this process is highly specific. The surrounding environment does a really good job of controlling the development of cells. And so what type of communication do you think is going on here? Chemical communication and local signaling. A lot of paracrine signaling, where you've got these signal cells, the inducers, that are releasing chemical signals that the responders are binding, transducing, and then executing a response. And then finally, determination. Oh, actually, that's not finally. It's the second to finally. Determination. And so after this early point of development, totipotency ends. Totipotency means the ability to become literally any cell type. So when that ends, you have what's called pluripotency, where it can become many cell types, but not any cell type. And then finally, you get differentiation, where now the cell starts to cement what genes are going to be transcribed in that cell, and basically takes on the role of whatever cell type it's going to become. So if that cell is going to become a liver cell, it transcribes or sets in the liver cell genes. Okay? And so all of this, it sounds nice. You're like, okay, the external environment tells the cell what to become. But what's determining the external environment? Other cells. Well, mom is providing the ultimate external environment, but other cells in the embryo are providing that external environment. So we have a question that we need to answer. Well, how do genes actually contribute to this process? How do genes contribute to this process? So here, just showing, summarizing this process, here we have a totipotent cell, okay? Here we have a totipotent cell. It goes through a process of determination and now is a pluripotent cell, okay, that can become several different cell types, but not any cell type. And then through this process of differentiation, will lock in the genes that it's going to transcribe and will result in it becoming a very specific cell type. And all of this happens as you get differing levels of gene expression, okay? Differing levels, so you make different amounts of certain protein products and you make different protein products. So summarize this real quickly. We have what are called master control genes. Master control genes, that what they transcribe is not a typical protein, but instead they transcribe and are translated into transcription factors. And what do we know about transcription factors? Somebody tell me. Somebody remind me what a transcription factor does.
It binds to the DNA.
Yeah, it binds to the promoter and recruits RNA polymerase, right? And these transcription factors are promoter specific. So if these master control genes are transcribing transcription factors, they are basically controlling which transcription factors are present and therefore which genes are going to be transcribed. So this process is controlled by induction in most cells. And again, induction is that process of the chemical environment telling the cells what to become. And then what you have is within the embryo, you have, say, at the very posterior end of the embryo, at the tail end of the embryo, you have certain chemicals that are being released by inducers, by those cells that are releasing chemical signals. Okay? And so the further away you get from the posterior end, the less concentrated that signal is. That make sense? If the cells that are releasing that signal are at the posterior end, the further you get away from the posterior end, the less concentrated that signal is. And so it basically creates a concentration gradient, where at one end, say the posterior end, the concentration gradient is such that the concentration is high there. At the anterior end, the concentration is very low. And so the cells basically know where they're at in the embryo based on the concentration of those chemicals. And then you have other chemicals that, say, are being produced at the dorsal end, the back surface of the embryo. And so as you get closer to the ventral surface, the front surface, the concentration drops. So now the cells know where they are on this axis based on the concentration of materials released at the posterior end. They know where they are on this axis based on chemicals that are being released on the dorsal surface. And they know where they are on this axis based on the way in which cells around them are developing. So the cell knows basically exactly where it is in the embryo, and therefore it's basically being told how to develop to get to that point where determination is done. It's kind of got a set range of cells that it's going to become. So the myoblast cells in the previous slide, they are capable of developing into any of the muscle types. They can develop into cardiac muscle, skeletal muscle, circular muscle. Well, what told that cell to stop being totipotent and become a myoblast, where it can only become muscle cells, what told that cell to become it is where it was developing in the embryo. And then once that happens, what tells it which type of muscle cell to develop into is a lot of still more external signals, internal controls. Does it make sense? And so I like to structure the class this way, that we finish up in this chapter 16. And we kind of bounced around a little bit. We talked about DNA replication and gene expression, and those were chapters 14 and 15. And then we went back and talked about, okay, well, now let's talk about cell cycle and talk about production of gametes. And then we went and talked about inheritance. And then we come back and say, okay, well, how do we control which genes get transcribed and how much? Because that's what facilitates all of these other factors. When do you go through the cell cycle? How do you execute the cell cycle? Okay, how do you make gametes? How do you pass on genes to the next generation? How do you create that division of labor that we know in multicellular cells? Multicellular organisms, multicellular cells. That makes no sense. Okay. Any questions? That's all I have for you. It's been fun. It's been real. It's been real fun. Okay? I will see you all on Monday. And yeah, hopefully we'll have a good time together on Monday. Monday at 11:30, same place. Almost the same time.
Monday, 11:30.